Hello students, I'm going to be reviewing the cell cycle and the stages of mitosis. Hopefully this material is somewhat familiar to you if you've taken a biology course previously. So I'm going to cover this in a fairly short time frame just as a review. So the cell cycle refers to the life cycle of a cell, and usually when people think about the cell cycle, they automatically think mitosis. Mitosis is part of a cell cycle. Not all cells undergo mitosis though, and the cell cycle is really a lot more than just the mitotic phase. So the cell cycle is essentially the lifespan of the cell itself. You can see here a nice little circular representation of the cell cycle, and you'll notice the portion of this circle that's in orange is the mitotic phase. So that's when a cell would undergo the process of cell division. And then the green portion is all of what's known as interphase. So interphase is the non-dividing part of the cell cycle. And like I said, usually people kind of confuse mitosis and the cell cycle for the same thing, but they're really not the same thing. So G1 phase, the first gap phase, you can see that in the purple slice of the pie here, that accumulates material that's needed in order to replicate the DNA. DNA replication, of course, is just making identical copies of the DNA molecules. That's going to be followed by S phase. S stands for synthesis. So we're synthesizing DNA in this part of the circle here. That's the DNA replication. And that, of course, has to precede the mitotic cycle. So for a cell that does undergo mitosis, it has to replicate all of its DNA in order to successfully partition the DNA into two identical daughter cells. That S phase is followed by G2, or the second gap phase. This is when centrioles are replicated and enzymes are synthesized in order to successfully divide the chromosomes and the cell itself. That finally is followed by the mitotic phase. The M phase, or mitotic phase, is nuclear and cytoplasmic division. Technically, mitosis refers to nuclear division, and cytoplasmic division is known as cytokinesis. And then in G0 phase, cells have left the cell cycle. They're not actively dividing anymore. And then the cell cycle duration is going to vary between cell types. So, some cells have pretty quick turnover rate, like our skin cells, for example, the cells that make up the lining of our stomach. Some cells, once they're mature, they don't actually undergo mitosis at all, like neurons within our central nervous system. And then there's everything in between as far as duration. So that is a quick review of the cell cycle. So mitosis and cytokinesis is defined as one cell's duplicated genetic material divides, that is mitosis, and then the cell splits into two daughter cells, that is cytokinesis. Functions of mitosis are embryonic development. So of course, all multicellular organisms that arise from sexual reproduction start out as a zygote, a fertilized egg. And that one single cell has to divide again and again during the course of embryonic development to give rise to, a multicellular complex organism. So mitosis is going to be critical for embryonic development, also for tissue growth, either during or after embryonic development, replacement of dead cells. Again, a lot of our cells don't live nearly as long as we as a whole organism do. So once a cell has completed its life cycle, it will die and be replaced by new cells. And then also repair of injured tissues. So if cells are injured beyond repair, they will need to be replaced. So the phases of mitosis, again, this is defined as the nucleus dividing, so nuclear division. prophase, metaphase, anaphase, telophase, PMAT. Timing of cell division is going to be important because, typically healthy cells that are not cancerous are only going to divide during certain circumstances. They have to have enough cytoplasm to partition into those two daughter cells, the two genetically identical cells known as daughter cells. The DNA has to have been replicated. There has to be an adequate supply of nutrients, growth factor stimulation, open space due to neighboring cell death, and cells will stop dividing when loss of growth factors or nutrients occur, and through the process of contact inhibition, which just means they are in contact with other cells. There's no room for cell division to occur, so they do not have to undergo mitosis to take the space of a cell that is gone, for example. Okay, so with mitosis, which again is nuclear division, and oftentimes cytokinesis is discussed at the same time since it occurs right during the last phase of mitosis or very soon thereafter. Mitosis will begin with prophase. This is when the chromatin, the loose thread-like DNA within the nucleus of the cell, is going to condense into recognizable chromosomes. And then the nuclear envelope that surrounds that DNA is going to disintegrate. We have spindle fibers that are going to grow from the centrioles, which aren't found in all cell types, but most animal cells will have the centrioles. Those centrioles are going to migrate to opposite poles of the cell, and the spindle fibers that are attached to them are going to attach to the chromosomes and basically orchestrate the movement of chromosomes throughout this process. Metaphase is when the chromosomes are going to align along the center of the cell. Spindle fibers attaching to those kinetochores, the central region of the paired chromosomes. And then the fibers of the aster coming out from that centriole are going to attach to the plasma membrane. Third part of mitosis is anaphase, when the chromosomes begin to pull apart from one another. So the centromeres are going to divide in two. Those centromeres are the region where the paired chromosomes were attached at the waist. The spindle fibers are going to pull those individual chromosomes towards opposite poles of the cell. And some of those spindle fibers are also going to grow in length and actually lengthen the cell itself. And then finally we have telophase, which is when the chromosomes are gathering at each pole of the cell. The chromatin will basically decondense, so we'll have this more diffuse thread-like genetic material, and then a new nuclear envelope is going to form around each set of genetic material. So that's telophase, and then cytokinesis, the division of the cytoplasm itself, which is of course all of the contents of the cell outside of the nucleus. Cytokinesis is going to pinch that cell in two to form our two daughter cells. So what we have to remember with mitosis is, of course, before this even started, DNA replication occurred. Every single chromosome was duplicated. And when those chromosomes separate, you have two complete sets of genetic material, such that you'll have two identical daughter cells. All right, so I'm going to ask a question and let you think about this one. A cell that must produce large quantities of enzymes would probably have a lot of which type of organelle? So think about what type of large biological molecule enzymes are, and then think about what type of organelle would be responsible for producing that. And then which of the following is true regarding mitosis? Again, think about that. And let me know if you have any questions. This is going to wrap up the week 2 material for the class, and please let me know if you have any questions.